All right. Welcome, everyone. I'm really glad you could join us today. I'm really excited for this one. It looks like it's going to be a really great presentation. I'm going to give you guys keyboard and mouse control. Hold on one second. Let's do that. See if you can move this. Yep, perfect. I'm going to take it back for 2 seconds. I'm just going to turn my camera off. So I'm going to kind of back away, but usually what I end up doing is I'll ask the presenters to kind of introduce themselves and to explain why they got interested in studying things like Chiari, and in this case, EDS. But with that, I'm going to turn off my camera and kind of let you guys introduce yourself and go crazy.
Okay, I'll let Cortney go first.
Hi, my name is Cortney Gensemer. I am a PhD candidate. I'm a fourth-year student in the Norris Lab. I am actually a patient with EDS myself, and so this project for my PhD has been super important to me personally. And so upon joining the lab, that's sort of how the lab shifted into the area of EDS research. So I'll let Chip talk a little bit more about that.
Yeah, so I'm Chip Norris. I'm a tenured full professor at the University of South Carolina. I've actually been here for a really long time, 27 years. I did my PhD here, 2 fellowships, really focusing on genetics and molecular biology, connective tissues, and started my lab in 2007, and our lab took a different direction after Cortney joined. We were really excited to see where we could go and see if we can make an impact for patients with EDS. I think that's us.
Yep. So, can we do it? Oh, there's me.
Oh, there's. So there must be a delay in the. Yeah. So just a quick primer about the lab. I really wanted to create a lab environment that could accomplish a lot of different things, and trying to understand how diseases are caused, how they progress, and is there anything that we can do about them. And I've studied over the years, I've studied both common and rare diseases, and have really recently focused, really because of Cortney, on Ehlers-Danlos syndrome. And as you see the picture, this is my group. It's a fairly large group. It is the largest group that I'm aware of in the world studying connective tissue diseases with a focus on Ehlers-Danlos, on hypermobile type. And so I think we're, what you'll see through some of the presentations are some of our successes and where we're going, where we've come from in really just a very short period of time. Really over the last 3 years, we've, I think, made a lot of significant progress on understanding EDS. And really the ultimate goal is, 2 goals of the lab are to understand mechanisms that cause altered biology and whether or not this can lead to pharmaceuticals or ways that we can actually harness the knowledge for patient benefit. Can we do something for the patients based on our understanding of what we uncover in the lab? And the second is really, I think, is a longer term vision, and I think it's something that's so critically needed, not only for EDS but for other rare disease, other diseases. I don't view EDS as a rare disease, but it's to develop a center that really brings together clinical and basic sciences to be able to provide a holistic care for those with EDS. It's a complicated disease affecting many different organs, and it requires a lot of different specialists. We should be creating centers that know about this and can really bring together physicians to connect the dots. And I think by doing that, the patients are going to do better. We'll be able to diagnose earlier. We'll be able to get them the care that's needed. And so this is really an important mission of my group, and is also becoming an important mission of MUSC. So with that.
So I'm going to talk to you guys a little bit about EDS. I'm sure most of you are familiar, but the Ehlers-Danlos syndromes are a group of inherited connective tissue disorders. They're characterized by things like joint hypermobility, skin hyperextensibility, and tissue fragility. There are 14 subtypes that are shown here, with the current 2017 nomenclature, previous nomenclatures. The inheritance pattern, so autosomal dominant is shown as AD here, and autosomal recessive as AR, and then the genetic basis of these types of EDS. And what you'll notice here is that all of the subtypes except for hypermobile EDS have a known genetic marker, which means that all of these genes have genetic diagnostic tests that are available to test for them and diagnose them at any point without needing a strict set of clinical criteria. So our research has been focused on hypermobile EDS in the lab. It's the most common type of EDS, although the exact prevalence is not quite known. We tend to go with a number of 1 in 500. And as Chip said, we don't consider it a rare disease. It is a lot more common than it's really acknowledged to be. Probably much more common than 1 in 500. And hEDS involves generalized joint hypermobility, a lot of musculoskeletal manifestations as a consequence of ligament laxity. So that can mean things like dislocations and subluxations, joint and spinal instabilities, the secondary osteoarthritis that develops as a result of this, and for most patients, a lot of chronic pain. It can affect any and often multiple joints in any individual. There may also be some mild skin involvement, so things like stretchy skin and poor wound healing, especially from surgeries and medical procedures, abnormal scarring, and then systemic manifestations such as abdominal hernias, pelvic organ prolapse, dental crowding, GI manifestations, Chiari malformation, which is pictured here, spinal and cranial, didn't realize the mouse did that. Spinal and craniocervical instability, and then mitral valve prolapse, which is actually something our lab has spent a lot of time researching previously. And then you might already know this, but there are some common associated comorbidities, which we're going to talk about a little bit more in our patient population that we have. But postural orthostatic tachycardia syndrome, or dysautonomia, or POTS, and then mast cell activation syndrome. The important thing to point out is that hypermobile EDS really is a spectrum of severity. So we see patients that are mildly affected and patients that are very severely affected, but we often see that women present more severely than men, and that the phenotype of hEDS tends to worsen around the time of puberty.
Which I think is actually giving us some really important clues as to how the disease really begins, and we'll talk a little bit more about that.
So we have established a patient registry, which we started in December of last year. And in that time, we now have over 1,500 participants that have reached out to enroll. We have 4 clinical research coordinators that are, 5 clinical research coordinators now that are consenting patients every single day. And we are collecting saliva samples from those patients for DNA analysis. We have collected patient demographics, medical history, saliva samples, as I mentioned, from 549 individuals with hypermobile EDS so far. And we are going to talk a little bit more at the end about how you can enroll for those who might be interested.
And I think just on the registry, this was really telling of how engaged the EDS community is. I was shocked at when we started the registry. I thought I was going to get 50 patients, and in one day we got 1,000, or roughly 1,000. who registered interest. And, you know, to collect this many participants is really remarkable in a short period of time. I think this is, I've been involved in, I don't know, a dozen genetic registries and genetic studies over the past 25 years, and this is just amazingly fast for getting this many patients enrolled. And it's great because it allows us to move very quickly into the next phase of the study, which is doing the more detailed genetics, which we'll talk about a little bit.
Yeah, and I also just want to add in here while we're talking about this, if anyone who's watching has reached out to participate and hasn't heard anything back, these are the numbers we're looking at right now, but we are doing our best to get back to everyone as quickly as we can, and our coordinators are working really hard to get back to you. And
also on that, I read every email that you guys send in.
He really does.
So it's because everybody is important. And so, and people want to get their saliva kits, people want to get, you know, their consents done. I understand that, and we're moving as fast as we can. But it's, you know, it's great to see how interested the population of EDS is wanting to be involved.
So I'm going to talk to you a little bit about the 549 people we have consented so far, and some of our breakdowns on what we've looked at in them.
And so, sorry.
Go ahead.
So I keep interrupting. So this is all new data. This has never been presented anywhere else. You will not have seen this anywhere else. You will not have heard of this anywhere else. This is from our registry. It's not from the EDS Society. It's not from other smaller groups. So, you know, I think it's a good cross section of what is real in the EDS community. Since we have the largest registry in the world now, this is the best estimates of the comorbidities and the phenotyping. And so I think this is really telling. Sorry.
So the first thing, looking at family history.
I'm just gonna interject for really quickly. So sorry, Courtney, can you speak a little slower because you're so far away from the mic? It's a little hard for everyone to hear you. Okay, and go ahead.
All right, let's scoot in. Go.
Okay.
Go, you can scoot in.
Sorry
I can be in the background. That's where I normally am anyway, so go, scoot in.
If you can't hear me again, feel free to let me know.
Is that better?
Sorry, the chat thing is going. Maybe people were trying to tell me that. Oh, I'm so sorry. I see the chat. Thank you for letting me know. Okay, so a little bit on family history. One of the things we're very interested in is families with hypermobile EDS, and we'll talk a little bit more about why later. But we found in our registry of patients that 80% of our participants have a family history of hEDS, which really highlights the importance of investigating genetic causes of hEDS in these families, and really shows support that this truly is an inherited genetic disease. Okay. So another category we looked at is sex, and one of the things we saw right away in our patient registry is there are a ton of females in our registry. So right now the numbers are at 89% as female and 6% male, with 3% answering to other. And we found this really interesting because along with seeing a higher incidence of females being affected or females having more severe symptoms, we also noticed that a lot of patients describe a worsening or an onset of symptoms around the time of puberty. So this is really brought into the picture looking at the potential role of a hormonal influence on hypermobile EDS. And it actually really fits with some of the genetic findings we have so far, which we will be talking about a little bit later. The next graph here shows a closer look at hypermobile EDS phenotypes. So specifically, some of those things that are in that diagnostic checklist or diagnostic criteria for hypermobile EDS. If you've never seen that checklist before, it is on the EDS Society website, and then it's also linked on our Norris Lab website that we'll have posted at the end of this presentation. We found that 22% have mitral valve prolapse, with 18% having other heart valves affected. We saw that 59% had poor wound healing, 67% with stretchy skin, and 71% with abnormal scarring, which I thought was a pretty high number for that one. 24% with abdominal hernias, and 20% with pelvic organ prolapse. So these are just some of the things that we were collecting that are on that diagnostic checklist for hypermobile EDS. But we wanted to look at other comorbidities that aren't necessarily taken into consideration for diagnosis, but that are frequently seen in patients with hEDS. Back to the criteria for a second, I forgot about this one. We also looked at self-reported Beighton score in these patients. So that is the 9-point scoring system for an hEDS diagnosis to look at hypermobility. So, you know, the elbow to your arm, pinky back, do your elbows hyperextend, do your knees hyperextend, all of those things. So, and this is self-reported, and there is going to be variability with age, but you can see the breakdown of sort of where patients felt. It was interesting that no one reported a Beighton score of 3, but we did have people through all parts of the spectrum. When it comes specifically to joint symptoms and pain, I expected high numbers in this, but the chronic pain was really higher than I imagined. If you're looking at 1 in 500 people having hypermobile EDS, what is that number of people worldwide? 15 million. 15 million estimated people with hypermobile EDS in the world, and almost all of those people have chronic pain. 89% have joint subluxations, and 62% have full joint dislocations. So that idea that hypermobile EDS is just being bendy is clearly not the case here. And then looking at comorbidities outside of the typical criteria for hypermobile EDS. So we saw that dysautonomia or POTS was reported at 68%. Oh, can you see this?
Can we see that? Can you guys see the pointer?
Yes.
Cool. Okay, so 68% with dysautonomia or a POTS diagnosis, and these are clinical diagnosis. So these aren't, you know, oh, I think I might have POTS, but I've never been tested for it. It's very specific. Have you been clinically diagnosed with any of these issues? We saw mast cell activation syndrome at 40%. Gastrointestinal manifestations was actually a lot higher than we expected. 81% of our participants have GI manifestations, so anything from IBS, GERD, gastroparesis, any sort of GI involvement was very high. We saw autism spectrum disorder at 9%, anxiety and depression at 69 and 75%, which is very common in chronic pain conditions, but also I think very telling of the experiences of a lot of patients with EDS and not being able to receive help or find answers or physicians for their various issues. With ME/CFS, we saw 60%, which was also much higher than I anticipated for that one. Am I reading the right one?
Yeah.
Okay, I looked misaligned. The sound is still pretty garbled.
Can you guys hear us okay?
I'll just go a lot slower. With adrenal dysfunction, wasn't aware was super common in hypermobile EDS, but something that I've heard patients have brought up before. So we did see a decent percentage of patients. This is 4.0%, not 40%, I believe. And then Raynaud's phenomenon was at 37%. Sorry, just.
Is it not going?
Okay, there we go. And so since this is a CSF meeting, I did want to touch a little bit on specifically neurological and neurosurgical comorbidities in hypermobile EDS. So we saw that 32% of our participants had upper cervical instability. So a clinical diagnosis, maybe even have had surgery of CCI or AAI, which was pretty high. We also saw that 9% had Chiari malformation and 9% with tethered cord. So we wanted to look and see in our data so far, do we see any correlations between certain things? Does your Beighton score, your degree of hypermobility as measured by the scoring system, correlate with chronic pain in our patient population? And the answer was no. We found no statistical difference between the groups, meaning that a higher hypermobility score did not necessarily mean that you were more likely to have chronic pain. And we also wanted to know, does your degree of hypermobility correlate with the number of comorbidities a patient has? And we found that it did somewhat. So we found a weak correlation between Beighton score and the number of comorbidities patient had. With higher Beighton scores, they were somewhat more likely to have comorbidities, but there wasn't a strong correlation, which is really interesting. So, you know, it's just really telling us that whether you have a 9 out of 9 score on the scale or you're lower on the scale with hypermobility doesn't mean that you might have more or less severe symptoms related to your EDS. So to talk a little bit about our focus in the lab, and Chip did touch on this, we're really focused on starting from patients and looking at the genetic causes, taking that and putting that into animal models, which give us a really nice platform to study mechanisms of the disease, to study diagnostic tools, and to study treatments for hEDS that can then go back into the patients. So a little bit on what we've been working on in the lab. Go ahead.
All right.
Make sure you're loud.
Yep. All right, so I'm gonna take over here for a little bit. That's all right. We can rearrange ourselves. There's a chat. All right.
Don't worry. No, that was fine.
Okay. All right. So we wanted to give everyone an update on what we've been working on and where we're going. And so this study really initiated with one large multi-generational family. So that means that there are a large number of individuals throughout the family. So great-grandparents, grandparents, parents, daughter, granddaughter, that sort of thing. It was a 4-generational family. And we wanted to take this, we knew that other groups were working on genetics for hEDS, and we didn't want to simply replicate what they were doing, and because it had been unrevealing. So we went with a different approach, figuring that if we had one very large family, it could tell us potentially one gene. And so we set out to do the genetics on that one family. And it took us about, I don't know, a year or less, I guess. 6 months. It was actually a fairly straightforward study. We did identify a very strong candidate gene. We're calling it a candidate gene because it hasn't been published yet. And if anybody has questions on that, I'm happy to answer them. It's a question we get all the time. We are getting very close to finishing the the data for the paper, which we will show you some of that data. We used a technique called CRISPR-Cas9. It's a technique that the inventors of just won the Nobel Prize for. And I guess maybe there's a question about what is CRISPR-Cas9. We'll address that a little bit later. And we were able to put the mutation we found in the affected patients from the family into a mouse. And so we were actually able to put the human mutation. It was a single base change. It was a single piece of the DNA that changed. And we were able to put that into a mouse. And we did that so that we could study how to understand the disease mechanisms. It also served the purpose of validating whether or not the mutation was causative to the disease, because if the mouse got the disease, and this was the only thing that we changed in the mouse, then we would be very confident that we were correct on our human genetic study. Importantly, the mouse, and I get that a mouse is not a human. I've been reminded that of many times. But a mouse is really the best way for us to, in the lab, to study how the disease progresses and to use it as a diagnostic tool and a platform for developing therapies that can then be translated back to the patients. So yes, the mouse is not a person, but it is very genetically similar to humans. Then we're also doing whole exome sequencing. That's another complicated term. We're doing a similar approach that we did for the large family on additional patients and other families. Since our initial strategy worked so well, we're hoping that it will continue to work well and provide additional genes that are causative. I click here?
Yeah.
Okay. Yeah. So we're calling the gene X. A lot of people want to know what the gene is, and I get it. But for right now, we have to we're unable to disclose the gene name. It will come out in the next couple. Well, we're going to submit for publication in the next couple months. So, hopefully we'll be able to release what the gene name is. But what you're looking at here is skin in the mouse. This panel here is the control animal. This panel here is the one with the mutation. The green, so this is all the connective tissue in the skin. The green is represents where the protein that's made by that gene is present. And what you notice if you compare the green amounts of green here to the amount of green here, there's much less. So this gene has a big problem with making enough of itself to get out into this tissue. Now, collagen is a major protein that's in the connective tissue space. It's what a lot of patients with EDS, EDS is thought to be a defect in collagen. We now know that some of the function of this gene is to have an impact on collagen. The gene we found is not a collagen gene, by the way. I guess we can tell them that.
Yeah.
Well, I just did. So it's not a collagen gene, but it does.
That'd be too obvious.
Yeah, it does have an important role in what collagen does. And so, if you look at the red, if people can see the red, this is the control, and this is the mutant. There's much less of the collagen. So what does this mean? This means that the connective tissue is not as strong and ends up being more elastic. So could explain why some patients with EDS have stretchy skin. We see the same thing in tendons and other connective tissues. We've tested the strength of the tissue. I'm not going to go through what this graph is. It's a way of mechanically testing how strong the tissue is. You basically put your, if you can see my fingers, you can put your little tissue between two clamps, and then the clamps separate. And when the tissue breaks, it monitors how far that tissue stretched. And so that's represented in these two graphs. In the end, this is the control, this is the mutant, and this actually tells us that the mutant is not as strong as the control and is more elastic. This will be maybe, is this the last data slide? Maybe there's one more data slide. This last. So this is actually looking at collagen. This is, I'm sure, probably the very first time that anybody on this call has actually seen what collagen looks like. This is taken from a transmission electron microscope. Electron beams are shot into the tissue, and what comes back is, based on a certain stain, is the actual collagen fibrils. So if you can see my little pointer, this is one collagen fibril. You need a lot of these to make your tissue very strong. And this is the control. You can see that they're about the same size. This tendon?
Mm-hmm.
What did I say?
No, you didn't say what.
Oh, yeah, this is in your tendon, Achilles tendon. So these are about the same size. And so this tissue is going to be really strong. When you look at the mutant, though, what you see a lot of these little guys. The smaller the fibril, the weaker the tissue in general. So why am I showing you this? I'm showing you this because it's validating that the mutation we identified is having an impact not only on collagen, but on the mechanical properties of the tissue and the stability of the tissue. And since we found this mutation in a family, we're fairly confident that this is positive. We always try to back up our genetic findings with biology. I think that's so critical. So there's a lot more data that we're not going to talk about. It gets, as you can imagine, very complicated. But we're going to be submitting our current work for publication soon. This will hopefully be going into a journal called Nature, and it's one of the highest impact papers. This will be really a monumental step forward for EDS patients and EDS research, and hopefully will be a springboard for other groups to really jump on now that we now that we're making good progress. Since we have such a large population, a wonderful population of EDS patients in our registry, that allows us to do large-scale genetic studies. So looking at what we have now is we have one mutation, one family. We now have additional information about this gene family and know sort of relatively how many mutations are represented in the EDS population. But we wanted to cast a wide net and ask the question, what is common in 1,500 patients with EDS? What is common in this group that isn't found in the population, of a control population? And so by doing that, we can find common features, common genetic variants that are causative. And so I think that will really round out and help explain the population genetics of EDS. And so that's what we're going to be doing in spring. We should have all of our DNAs for the 1,500 patients isolated by then. We are going to continue collecting. So we're not stopping at 1,500. If people want to be involved, we want them to be involved. We're not going to shut off, our enrollment just because we've made these discoveries. We want people to stay engaged, and whenever you finish these large genetic studies, you start on your next large genetic study. So it's going to be in perpetuity. So we want people to stay engaged. And then we're going to look into researching comorbidities and how they relate to EDS, POTS, mast cell activation syndrome. Our mice actually do have, appear to have mast cell activation syndrome. It's the only model that I know of where we have hyperactive mast cells based on a genetic mutation. So it's really exciting. And then also, how do hormones potentially play a role? As Cortney said, the sex bias being almost 90% in female, and the sort of initial symptoms of the disease initiating around puberty really strikes us that there's going to be a hormonal influence. We do know that our gene, the gene that we've identified in the gene family that we've identified, is hormonally regulated and is produced by the adrenal gland, which is a major hormone producing glandular tissue. And so we're very excited about looking at how hormones are regulating the disease. You want to hit this?
Sure. So let me get in here. Yeah. So I think a lot of this, a lot of this kind of stems from, I think, me also being a patient myself, and knowing how ignored and neglected EDS is in the medical community. And so as part of our research efforts, we're really doing our best to also educate and do outreach and awareness and advocacy for EDS as a disease in general. So one of our first things we did when we were starting our research is we wrote a literature review, and the goal of this was really to take all of the information that's out there on hypermobile EDS, everything that's been published, and take that information and package it into one simple paper that can be understood by patients and physicians and other scientists, that covers everything from the diagnostic criteria, the symptoms, the comorbidities, and then the science that exists so far. So when it comes to that science that exists so far, it's pretty limited, but there is some limited information out there. And so we kind of took this as a stepping stone to say, there needs to be more research. This is us kind of getting ourselves into the field of EDS, and we're going to continue to work on this. What's really cool is we recently learned that the literature review, which was published in a journal called Developmental Dynamics, is actually the most downloaded article in the history of the journal. So that's really exciting for the community that people are reading this. 120-year history. 120-year history of the journal, the most downloaded article. So, it really shows that patients, they want to learn more. People are probably taking this to their doctors, or doctors are sharing it with each other, which is really, really awesome and impactful. We're also obviously trying to get involved with various societies. So doing this today with the CSF, also working with other societies like the EDS Society and other groups to really be involved and be a part of the global research community on EDS and comorbidities. We also hosted a MUSC awareness and fundraising event with Miss America 2020, Camille Schreier. There was a really great article published by the MUSC Catalyst, and all of these articles that I'm talking about are also on our website, which we'll share the link to at the end of this. But Miss America actually has hypermobile EDS herself. So this is an issue that's really close to her. And as a woman in science, as a pharmacy student, what we were doing in the lab was even more exciting. So she came here and really helped put our research and hEDS out there and on her social media platforms and interacting with people at our university to really get EDS more on their radar, which was really awesome. The article that was written about it in the Catalyst is actually the most read article of any MUSC Catalyst articles they've ever published as well. So it's really awesome to know that people are so excited about what we're working on.
Yeah, no, go ahead.
Where's the picture? Oh, there it is. So the last part of our outreach and advocacy efforts, which I think is the most important and the most impactful, is our hypermobile EDS internship program, or HIP. So we started this this past summer. It was our first summer doing it, and it's an opportunity for undergraduate, graduate, medical students, or postdoctoral students who have EDS themselves that are interested in studying the disease and want to pursue a career in science, research, medicine, healthcare, genetic counseling, anything like that. And so we brought in 4 interns this summer. We actually had a 5th intern through another program. So we had 5 interns all working on hEDS in the lab this summer. But not only did they get to be a part of the research, they also got to participate in other things like shadowing with Dr. Sunil Patel, who's also at MUSC, who you guys, I'm sure, have seen on here several times before, and getting to interact with a lot of patients and get that clinical perspective as well. But our bigger goal with this is really to build a group of physicians and researchers and nurses and physical therapists and genetic counselors who understand EDS and who will go on into their careers and teach it to others. And so as part of this program, we hope that we build this network of physicians and people who understand the disease that will eventually start shifting the perspective in the clinical community, so that when you're taking your kid to the doctor and they're hypermobile and complaining of pain, a doctor recognizes those signs right away. So we plan to continue to do our hEDS intern program every summer, and it's just a really cool opportunity to have other people who are so passionate about the disease be a part of our discoveries in the lab.
Yeah, so I mean, starting, you know, it's awful to hear patient testimonials about how long it took to be diagnosed. 14 years on average. And that's unacceptable. But how do you change that? How do you change the perception of the physician? And it's a challenge. And you can do awareness, outreach, advocacy, but we're taking the approach that we want to start at the ground level. We want to start before these physicians become physicians, before the clinicians become clinicians, before they become physical therapists or, you know, rheumatologists, or orthopedists, or neurosurgeons. We're starting when they're in college. And when they graduate from their, that was part of the requirement, the criteria for joining the program, was you have to want to do this for your career, either medicine or research or health professions, or some aspect that is going to help the EDS community. That was a priority. The second was that you had to have EDS yourself. And so by doing that, we're hoping that when they graduate from their professional college, that they will go on and teach the classes and teach the students, the 200 students, about EDS and the various comorbidities. And so I think it's our attempt. We're just one lab, but I think it's an amazing opportunity that we have, and so we're going to do as good of a job as we can with it. Everybody in our program this past summer wants to do different aspects. Not everybody wants to go into medicine. Not everybody wants to go do research. We have somebody who wants to be a genetic counselor. We have somebody who wants to be a nurse. Somebody who wants to be a medical doctor, and another person who wants to be a researcher. So by that, we're actually spreading the wealth, or spreading the knowledge. You know, I think it's really an important program, and I hope that other labs around the country will hear about it and will sort of follow our lead.
So before we get into the Q&A, I just wanted to mention, if you're interested in learning more about our lab, more about our intern program, or you want to enroll in our registry, our lab website is thenorrislab.com. Or if you're sitting here right now on your phone, you can also just scan this QR code and it will take you to our lab website. We have a volunteer who's just started in the lab who's working on keeping our lab website really up to date for us. So we have a news tab where we talk about highlights from the lab, and we'll constantly sort of be updating with what we're doing, if we've published anything, and all of that.
And I'm seeing in the chat that there's some that are interested in the internship, which is great. Check out we're going to open the call for the internship in February, I think.
Yeah, and we'll have all of the information in terms of criteria on there. It was an awesome group of applicants last year. It was not an easy decision. We had a lot more than we thought we were going to get, and there are an incredible amount of very talented and brilliant EDS students out there, despite living with a very challenging disease. So it's really awesome to see so much interest.
So I think
yeah,
that's it.
I guess we're ready to get started with some of the questions.
Excellent. All right, perfect. That was really awesome. Thank you guys so much. I have a ton of questions to get through today, so we're gonna try our best here. I know you talked about it a little bit, but I want to kind of go back to just like very, very basic stuff to just give some definitions about a lot of what this is. Really, like, talk to me like I'm 5, kind of what is EDS? So really quick, what is collagen? What does it do in the body? And what is it about collagen in people with EDS that makes it different? Why is it different?
So we view collagen as, well, Cortney actually had the best explanation of this when I don't remember who you were talking to, but it's the glue that holds you together. It's made by cells in your body.
It's everywhere.
It's everywhere.
It's in your skin, your bones, your tendon, your ligaments, your mouth, your, like, everywhere. Your eyes.
Not so much in your brain, which is interesting, but yeah, it's
the most abundant protein in the body. Someone just added that in.
Yes, yeah. And so defects in collagen are, you know, if you have a defect in that, it's going to affect a lot of different tissues.
So there are 2 kind of ways to look at collagen diseases. There are diseases of excess collagen. So when you think about fibrosis, when you have too much collagen, you get a very stiff tissue that doesn't want to bend and stretch a lot. When you have defects in collagen, or it's not formed correctly, or you don't have enough, you get really loose and stretchier tissue.
Yeah, and when you look at them under the microscope, they actually look like cables. They're like steel cables. So you can imagine sort of very small steel cables running through your body, and if you can't make those cables, then your support system for your bones and your muscles is not there.
And I think this is a really important question to kind of lay the groundwork about what you're actually looking for. So genetics and cellular biology is very complicated. So what does it mean when you say you found a gene for something? So really, what is a gene, and what does it tell the body to do? How does it work? I know you said that the gene that you are looking at, this gene X, is it's hormonally regulated produced by the adrenal gland. So like, what does that mean? How do you go from gene X to adrenal gland, and what does that mean for patients with EDS?
Sorry.
Go ahead.
Okay. Well, so it's really interesting. I think that if the gene that we found had come up from another genetic analysis from another group, they probably would have thrown it out and said, this is in the adrenal gland, it probably isn't relevant. It didn't. There are a lot of publicly available databases. Some of them aren't that great or that in depth, depending on the gene, but a lot of them just kind of showed that it was in tissues that didn't really seem that relevant.
So I'm going to back up here.
Okay.
So a gene
what is a gene? Sorry.
carries information that determines your traits. So it carries the information that makes you who you are, and you inherit it from your mom and your dad. One copy of each gene from your mom and your dad. So that's what a gene is in very simplistic terms. And so finding mutations is challenging to describe in layman's terms.
When we started the project, when we first got our genetic data back and we're looking at the family, we started with 48,000 mutations that we were looking at. And that's with two people who are related. So had they not been distantly related, it would have been a lot more. So you start with a huge list, and there are a lot of different ways that we work on narrowing that list down. So some of that can be things like looking at if the change is in a part of a gene that actually makes a protein. So that's when they say something in an exon or something in a coding region. So that's something we look at. We also look at how common is that mutation in the general population. If it's pretty common, it's not going to be causative for EDS. And then there are various different prediction algorithms and platforms that can give you some information if that single change might be damaging or if it probably wouldn't do anything bad.
So the answer I saw on the chat, there was a question about whether or not this was a coding change or a regulatory change. Kudos to the person who asked that. A great question. Somebody who must be very scientifically minded. So this is a protein coding change. It is a single base change that converts a glycine to an arginine, right? Yeah, glycine to an arginine amino acid. That actually completely changes the function of the protein. And as a result, the protein fails to get out of the cell. And then it gets broken down. So it's essentially what we call a loss of function, where it's protein's lost and it can't function. So it's a loss of function. Great question.
So back to the adrenal gland. We didn't finish that part.
Adrenal gland. Yeah, go ahead.
So we had seen that it was known to be expressed in the adrenal gland, and a lot of people would be like, that's not related. Move on to the next thing on the list. But it was the only gene mutation that actually tracked through the family. So every person in the family that had EDS had this mutation, and every person in the family who didn't have this mutation. And so we thought, maybe we need to take a closer look at this. And so we did some preliminary, easy experiments in the lab and found that it was in skin, it was in tendon, it was in the GI system, it was in every single tissue that we looked at. So that's when we were like, well, yeah, it's made in the adrenal gland, but it's also in all of these other tissues that are relevant to EDS. So the adrenal gland wasn't really something that we were ever looking for. It was something that we kind of found coincidentally. but was super interesting to us because it is the hormone producing gland.
Perfect. Thank you. Yeah, that was a pretty all-encompassing. There were a couple of questions about that, so I tried to get as many in as possible. So this candidate gene, is this a gene that comes up in a normal genetic panel? I know something like this, when you identify a gene that's causative, it takes a long time to go from the bench to bedside, so being used in the clinic. Is this like a common gene that you would be looking for in clinic, or no?
No. It's probably something most people have never heard of, honestly. And it's not something where it's not like associated with any other connective tissue disease. It's not associated with any other disease.
It's not on any gene panel.
No.
Yeah, so when patients with EDS go and get their gene panels done, this is definitely not one of them. And so it is a completely novel discovery. Now,
that's good.
we're often asked about how long, once we publish this, how long until it gets on a gene panel. It's a hard question to answer because we don't really set the timeline on that as scientists. I don't actually know who sets the timeline on it, but I do know that from my previous studies, some of the genes we identified are on gene panels, and it took a couple years, which is actually pretty fast.
For mitral valve prolapse, not for EDS.
Yeah, not for EDS, but for mitral valve prolapse.
I mean, but even one of our bigger goals would be to, work on ways of having a diagnostic test that can be, you go to your primary care doctor and they send you for blood work and they can test for EDS, and that something that's more accessible to patients than genetic testing and can be done quickly and a lot more easily and a lot cheaper too.
You guys have great questions, by the way.
Yeah. This one actually came in ahead of time too. So you talked about the mast cell activation syndrome kind of appearing in the mouse model that you put together. Is that also, are these symptoms, why do you think that was replicated in the animal model? And is that also something that would, I'm sorry, I'm so confused. I'm reading from two different screens. And is POTS also something that you noticed? I don't know how you would study that in mice, but I'm
little
curious.
tiny tilt tables. No, I don't know.
We have tilt tables, but yeah, studying POTS in a mouse.
It's gonna be one of the trickier things.
Is their heart rate is about 400 beats per minute or more, and their blood pressure, I don't know what their blood pressure is, but there are blood pressure cuffs for mice. You put them on the tail, they're tail cuffs. So you can do that, and we're trying to figure out what's the best way to do that. That's going to be a tough thing to monitor.
I think with the mast cell thing was sort of a let's look in the family that we identified the mutation in. There's a high prevalence of mast cell issues, so we thought let's just look for it. I did notice some weird skin things in some of our mice, like weird patches of red skin and things that just I was kind of like, this, we don't normally see this in mice. So the data on the mast cell stuff is pretty preliminary, but it is promising. We have to do more work to figure out how that's connected. Like, how is that connected to the gene that we found? How does it connect to EDS? Is it that the connective tissue allows mast cells to move more? Is it something else in a mast cell pathway?
Yeah.
We're still working on a lot of that.
Yeah, that's awesome. I'm trying to find a question that makes sense next. Oh, this is actually a really good one. I was curious too. So there's variation amongst all the types of EDS, especially even in hypermobile EDS. So there's variation in severity. So the question kind of was, could there be various mutations in people who maybe that results in that phenotypic difference? So there's more severe symptoms in patient A who has different mutations, and how far does this rabbit hole go?
I think it's a really interesting question, and yeah, I'm not going to be surprised. So we already know there's many genes, and I don't know if we can disclose how many we found, but we found more than one. But the link between the specific phenotypes is really interesting. Are there genes that explain a patient with EDS and POTS and mast cell? Are there separate genes that explain Chiari and EDS? You know, are there these sort of subheadings or subpopulations of EDS patients that should be considered, you know, almost a different disease if they're linked to a different gene? I think it's a fascinating question. We don't know. I won't be surprised if we find that we can sort of parse out different subtypes of EDS. But we'll have to see. The genetic study we're doing in the spring will really allow us to look at some of that, and I think it's going to be really exciting and really hopefully revealing of not only the complexities of the disease, but also how different genes can work together or work, you know, to control the connective tissues.
Kind of bouncing off that, and maybe make it a little simpler by explaining a complicated thing, that different things like height are caused by multiple genes. So you're kind of alluding to the idea that maybe hEDS or EDS in general might also involve multiple genes. So like, how does that work? How does something like that work? And how would more than one gene control something as complicated as hypermobility?
So there's like two sides to this. So there are diseases or even just characteristics that are controlled by multiple genes in the same person. And then there is the same disease that can be caused by individual genes in different people that are different, if that makes sense. So even looking at other subtypes of EDS, some of them might have two genes associated with them, but it's really one mutation in one gene in each person. So is it possible that some patients might have multiple mutations in different genes in just one person? I'm sure that's absolutely possible. However, in what we're seeing so far, it is one gene per person, but multiple genes together. Does that make sense? I don't know if that was a good way of explaining it.
Yeah, so I like to think of it as the building of your tendons or your ligaments or your bones does not rely on one gene alone. It relies on literally thousands. And disrupting something along that pathway, that's one gene that's involved in sort of initiating bone growth or tendon growth. You know, there's a lot of different genes that play a role together. It's sort of like if you build a house with bricks, every brick's important, and you take out a brick that's really critical for its structure, you know, then the house falls down. And so there are a lot of bricks that you can take out that won't cause a big defect, but then there are a lot of bricks that you take out that will cause the disease. So it's not going to be one gene for EDS. We know that already. We don't know how many there will be, but they may fit in the same pathway. So the function of the gene that we identified, maybe they're working together in concert with these other genes. So I think we'll have to see what happens, you know, with our further genetics discoveries.
So another, I hope, more simple question. It's really about the heritability. So there's a very blatant question: is hEDS heritable? Asking about whether or not a mother could pass it down to a child. And then there was a really interesting question that I don't know the answer to it because I am not a geneticist. How likely is it that two siblings with the same two parents could have genetically different types of EDS? Is that a likely thing? Those are two questions, really. Sorry.
So yes, it is heritable. And as we showed, 80% of our participants have a family history. That doesn't mean that mutations don't happen spontaneously. So we call those de novo mutations. So you could be the first person in your family, but you could absolutely pass that on to children. And then in terms of the second question, if there were two siblings to have two different mutations, it would be more likely that, and I don't know in that scenario, that both parents had a different mutation for EDS. So let's say in an example, the mom had classical EDS, the dad had vascular EDS. One kid got classical, one kid got vascular. It would be a very unlikely scenario, but it's absolutely genetically possible.
Yeah. If both parents are not affected, it's still possible.
Yeah.
But exceedingly, I mean, it's really, really, really rare. So yeah, but it is, yeah, it is possible.
Okay. I'm trying to pick a next one.
Oh, Kathy's and Kevin. Yeah, the arginine is an interesting one. It's actually glycine, not lysine. It's a glycine to arginine change. Huh? Aspartic acid. Oh, cheated D. I'm thinking. Sorry. We've identified multiple genes, and I'm thinking of the one of the other genes. It's a glycine to aspartic acid.
The other one is a glycine to an arginine in a different gene, so he's not wrong.
Yeah. Sorry. The one that we made the mouse of is a glycine to aspartic acid.
Okay. So we're talking about heritability. It's suspected. I read your review that the gene for hypermobility is potentially autosomal dominant. So is there a reason that it would occur more frequently in women? Because that doesn't really shake out when you think about it being 50-50 male-female at birth.
I think there's a couple possible explanations for this. The first being that in a lot of families, you find that the women in the family are the first ones to be diagnosed because their symptoms are more severe. So I wouldn't necessarily say that more women have hEDS. I think that women are more severely affected. It's also been shown that women are more likely to go to the doctor when something's wrong with them. Men tend to not. So more likely to get a diagnosis because they go to the doctor, they're more severely affected. Also, there has been some evidence that women are also more likely to enroll in studies and participate in things like this. So that could also be somewhat skewing the information. But I have heard in a lot of families that have men, they are diagnosed because their sister or their mom or their daughter has EDS. And then, yeah, so that was the case in my own family. Hi, Dad, I know you're on here. My dad would have had no clue that he had hEDS had I not been diagnosed. But then it was very clear, like it did not come from my mom, healthy as could be. And it was like, oh, Dad, remember how you tore all those things and all those things happened, how you could do splits? Like it made sense, but I don't think he would have ever known if, I hadn't been diagnosed. So I wouldn't necessarily say that there are more women who are affected, but women are more likely to be diagnosed, I think is a better way to put
it. So in the mouse model, were you able to determine a likely trigger for progression of severe symptoms in carriers of the candidate gene compared to those that carry the gene but appear fairly asymptomatic?
So mice are a lot different than humans. They tend to be pretty consistent if you do anything to them to cause a disease. So mice are like, there's no difference. The mice we use in the lab are genetically identical. They have no differences from one mouse to another. So they don't have any, aside from being male or female, there's the background on them genetically is completely the same. And so there aren't any other factors that can really influence symptom progression or severity aside from hormonal influence. So basically our mice are consistently affected to the same degree because they live in a cage and they have the same activity as each other, and they're not, it's not like one person's playing sports and another person is doing art instead. You don't kind of see the lifestyle impact that we see in patients. I don't know if you have more to add to that.
Yeah, I mean, our animals are genetically identical except for the one base change. Now we can, and it's a really interesting question because we can outbreed them onto different backgrounds, different strains, and so we can actually mix up their DNA, sort of like we all are, right? We're all different. That's why we look different. So it's an interesting question that I hadn't really thought about, and maybe it's something that we should consider doing down the road. So, but we can start to look at that by doing these outcrosses to different types of mice.
I know you kind of talked about this. There's a couple of good questions coming in that I want to save for the end, but is it plausible that some of the genes that would be identified will be missed in this initial first round of your study? And do you have any other candidates for genes, which I think you did cover already? So how many do you think you've missed so far, I guess?
How many did we miss? I don't think we missed any. In the samples that we've received, I think we in the family we certainly I think got that one, and then we took a wider approach in close to 120 additional patients we screened, and we have an idea of the genetics of about 20% of them, 15 and 20%. And so to answer your question, how many do we miss? We miss maybe 80% that we don't know the genes for. I don't like to say it is not as missed because I think we just haven't discovered it yet.
Yeah.
Right.
And so, but that 80% is obviously a very large population. We're going to capture probably most of that, a vast majority of that, in our larger genetic study in the spring. That's the way that we wanted to start was with a large family. And the mutation that we found in that one large family is going to be rare. It's not going to be shared by everybody. So that's not going to be a diagnostic tool necessarily. It does give us a model to study the disease.
And it gave us a lot of insight into what people haven't been looking at. So that like 15, 20% that we found is all because we started with that one family.
Yeah, and going from essentially one person to 20% of the population is a huge jump and a monumental discovery for the EDS community, but we want to get more, and so I don't think we've missed anything. We just haven't discovered them yet.
Right.
We haven't discovered all of them. I don't know if we're ever going to discover all of them, but hopefully we'll discover enough to really make an impact and transform people's lives, especially with diagnostics. That is a major driver of our research, not only developing therapies and treatments based on our discoveries, but having a diagnostic tool. A couple years ago, a paper came out that said 86% of patients with EDS, had they been diagnosed earlier, wouldn't have had such severe comorbidities and such severe problems. And 14 years to diagnosis is insanity. So we need a diagnostic tool. We need to be investing in large-scale genetic discoveries in labs that really have a focused interest in genetics and biology. And so, we're hoping to be that. I mean, I think we're doing a good job. We're being one of those labs, probably one of the only labs But I hope that other labs join us. We're just one lab, but we want more people to join us.
That kind of ties in. There's another question. Do you know about any plans to update the diagnostic criteria once this research gets published? I know we talked a little bit about it before, that there's no good timeline, but that's where organizations like ours and EDS Society comes in, to try to, push that forward as far as advocacy is concerned.
I think that if we, sorry, go ahead.
No, go ahead.
I was just going to say, I think, the question about, will there be certain genes and that kind of branch off into different subtypes? I think in a scenario where we see clear symptoms that align with specific genes or a specific gene, I would absolutely see that there could be revisions of, names of types and specific diagnostic criteria. But also, we're not clinicians, so we do get the question a lot about, like if we can help diagnose EDS. And although sometimes I feel like we could, we're not qualified to really make huge changes to anything clinical like that.
Yeah. There was an interesting question that the way it's asked, it's specific to the sacral canal and kind of talking about the area by the tailbone. As far as the ligaments being affected in hypermobile EDS patients. The question was, would sacral instability be an issue worth investigation as far as what you guys do is concerned? And the phylum terminale and like tethered cord syndrome. But I would even go bigger. So did you investigate instability in your mice? So did you do little MRIs and
we want to.
Do you want to tell them about how you extract them? Maybe
No.
not. Maybe not. Okay.
Mice are really tiny. So, and I know one of the questions that I got to was, how do you test mice for hypermobility? So this kind of fits in with that. It's really tough. And also, mice are super bendy to begin with. Even a normal mouse, I think that's how they fit in small spaces and do all kinds of crazy things. And then of course they're on 4 legs, so the impact on the spine could be completely different. We've found ways of, as Chip had talked about, sort of stretching tendon and stretching skin and looking at mechanical properties in terms of looking at hypermobility. We can actually tie their tails in a knot, which is another thing that's kind of interesting, because their tails are looser. We don't have tails, but maybe you'd be able to do that to us too.
The other thing is Cortney's handled lots of mice over her time in the lab, and when we first made these mice, she said the first time she picked one up, it turned its head almost all the way around and bit her.
Yeah.
So, and that's not, the mice try to bite you. They're not happy with you handling them all the time, or at least when I try to handle them, which is not very often. I
have never been bit by a mouse before. I'll just say that. Nothing like that has ever happened, and I just have never seen one turn like that. So we're definitely interested in ways of imaging and looking at some of those things. But as you know, assessing for craniocervical instability with imaging in a person is already very difficult. And so finding ways to look at a lot of that stuff in a mouse is going to be a lot harder.
Yeah.
Absolutely.
I know somebody asked about the percent of EDS, what percent of hEDS is explained by the gene. So it's hard to estimate. We've been doing more around the gene family. There are numerous members of this gene family, and that's the number that we threw out before, probably 15%, 15 to 20% from this one gene family. So that's about the best estimate that we have. We'll be able to know more in the spring with our larger patient pool. Sorry, I'm just looking at the chats, and I know that there's a lot of questions I feel like I should respond to, but if you have other questions.
So the other questions that came in ahead of time are either clinically related, so I honestly, I don't know if I would be able to ask them to you, or we've kind of already touched on them. So if you want to start going through the chat too, we can definitely do that.
So
I know
the question about
yeah.
the phylum outside the midline. So I sent some of those questions to Patel. And I
Okay,
said,
good.
I don't understand these words because I'm a scientist and you're a physician. So one of them was
The
about
the
it was
Susuda,
about,
yeah.
let's see, it was
3 out of 27 normal
yeah,
cadavers
3 out
had
of
an
27 normal cadavers had an initial had an offline fusion of the phylum to the dura in the sacrum. This kind of difference causes torque in the entire dural tube, ultimately having an important effect in the lower brain and upper neck. And so Dr. Patel said that dural attachments of the phylum in any atypical location, regardless of side or midline, does, I believe, in some cases, cause a physiologic torsion or scoliosis of the spine.
Okay.
So that was, there you go.
Asked and answered.
Yeah.
There was
Yeah.
another question that someone had sent to me that I just thought was something that was really important to touch on. And that question was, is it known why it has taken so long for a candidate for hEDS to be found? And I think we kind of alluded to this in a few ways. One of those being that there's going to be multiple genes. And so by starting with a family, we had the opportunity to take people who were related to each other that for sure would have the same mutation because we could track how it was inherited, take people who were distantly related, and then narrow down to something. If you take a bunch of individuals who might have mutations in different genes, and then you're comparing what they share, you're going to not really get anything back from that. So I think that's one reason. Another reason could be like we were talking about with the adrenal thing, that sometimes maybe biologically, when people are doing genetics, they look things up and the biology doesn't seem to make sense that they find online, so they might throw it out. I think we have the advantage of that we aren't really clinical geneticists, so we don't sit in a clinic and see patients. We have a lab that we're able to test all the biology in. And I think there's a really big disconnect. Patients go to a geneticist, they have a panel done, or they have whole exome sequencing done, and they say, we didn't find anything, or we found a VUS, a variant of unknown significance, which a lot of people get back. And that's it. It doesn't go anywhere. It doesn't go to a lab. It doesn't go to a group of researchers who can then test those things biologically. And so that kind of disconnect has also made it a little bit difficult. That's part of the reason why we're so interested in developing a center where there won't hopefully be any disconnect between the clinicians and the researchers working on the disease, because we'll all have that shared goal of overall better care for people with EDS and other connective tissue diseases.
Yeah, and we'll be very accessible. I would envision that we'd be very accessible. I mean, you know, with the question that Cortney posed, why is it taking so long? You know, another thing is we all have thousands, tens of thousands, hundreds of thousands of changes, differences in our DNA, and they're not all causing something, but when we get ourselves sequenced, you see this list of 100,000 SNPs, or single nucleotide polymorphism, you know, variants, and there's just no way to filter through that. And when you get that information and then you you try to filter it through by biology. So what are the rare variants, and then what is the biology? The gene we found, like she said, would never have been on anybody's radar because it's never been shown to be in connective tissues. The highest expressing tissues are the adrenal glands, salivary, thymus, like secretory glandular tissues. And that for us was extremely confusing and paradoxical. It didn't make sense. Like how could something expressed in the adrenal gland affect your affect your joints and your your cranial cervical spine? It just didn't make any sense because we didn't understand the biology until we started looking at it more. And it is that's the neat thing about science, is that you get thrown curveballs, but figuring it out is what's really cool.
Absolutely. Yeah, that's why I like science. Before we move away from this, there's a really good question in the chat, but I just wanted to ask this one since we're talking about adrenal. Obviously, yes, but are you finding any indications of adrenal or mitochondrial issues in your specific research? So in your mice model, I imagine are you seeing adrenal symptoms or mitochondrial issues?
Yeah, we haven't looked at that enough. Really, the first steps for us were identifying the gene or a gene, make the mouse, and then at least show that the mouse has phenotypes that are consistent, and then with phenotypes consistent with the disease, and then get that piece of work published. So the adrenal is a very interesting area that we're going to be spending a lot of time on.
We kind of get a lot of these questions. Have you looked at this? Have you looked at this? Have you looked at this?
Yeah.
And we think all of them are super interesting. But right now, showing the mouse has the disease and publishing this has been our top priority. And then there will be very many small projects, some of them digging in deeper into molecular biology and consequences in the connective tissue, and others maybe looking at mouse activation syndrome, looking at some of the gastrointestinal stuff. We're already, interested in, how do we look at gastroparesis in a mouse? And there's a lot of areas that we want to investigate, which is why we want to keep bringing in interns and students into the lab, so we have people to take on all of these projects.
Yeah, absolutely. And so I you saw the question in the chat, and I would even explode it out to other. Why did you use mice versus rats? Is there a scientific reason? I think I know the answer to this, but is it a scientific reason or some other?
First of all, hi Cheryl. Good to see you again. Do you want to answer why mice? Interesting.
They're cuter. I'm just kidding.
That's what you got?
No. I don't know if I know the right answer, so I'm gonna let you answer it.
So doing CRISPR, we could really choose any animal. We could do it in a rabbit. We could do it in a horse. We could do it in a pig. A horse would be a little.
I don't know
much,
where we keep those.
I don't know, yeah. But CRISPR has allowed us to do genome editing in any species. Mice were chosen scientifically. They weren't just chosen because they're cute. We have a lot of different mouse strains that will allow us to, we can mate our mice, our EDS mice, with other mice that have different genetics going on that allow us to actually understand more what's happening in EDS. It's a very complicated to describe, but we did it scientifically because it would actually, through breeding with other lines of mice, would be able to say what cell types are making this gene. If we can remove it specific, we can remove the gene specifically in the adrenal gland if we want to. So, but that is done in mice. We don't have the genetic tools in rats to do that. Yes.
We could have used rats, yes. Like we have done the CRISPR in rats, yes. But down the road, all of the other animal models we have for other things in the lab are also mice. I don't know if this is right, but I'm assuming they're also probably reproduce faster, They're cheaper scientifically to work with, cost less to house mice. Yeah, what else though? Oh, someone else just said that same thing. So it's
I was actually thinking that you were gonna say it was just ease of they're easiest to manage. They're small and
No, I mean that is true. That's but scientifically there is a scientific rationale for it. Yeah, Cheryl, I would love to have a larger animal model, and maybe maybe we'll develop one. I think we would go bigger than a rat. We'd probably go to a guinea pig, or I don't know, something bigger to be able to see something in the MRI would be great.
we have some contacts with some veterinary practices, so if you wanted to do some dogs, we could probably get that.
We did mindful follow-up stuff with dogs before. Yeah, not we didn't have the dogs, but with other people who had dogs.
Yeah,
at least clinically. Okay, so I want to end on like two different, more forward-looking questions. So, first of all, what does this research mean? How does this potentially change the lives of patients with EDS in the future? What do you think?
Yeah, go ahead.
Think big.
Go for it. You're the one with EDS. How is this going to change your life?
If we think like small scale, like paper comes out, there are genes mentioned in the paper, and you are looking for that tangible evidence that you have this disease, right? Because without genetic testing, you could get a clinical diagnosis by a doctor, walk into another doctor's office, and they'll try to tell you they don't have it. I mean, I've heard these stories from so many patients. Or, you probably don't have that, or that's overdiagnosed, or that's just being bendy. And this is going to be a really strong scientific paper that is saying that, like, this is a real disease. And I think that for patients is, one, going to be validating, and two, giving them a tangible piece of evidence that they have this. But like going beyond a first paper, it's going to bring more awareness, right? So if this goes to a really good journal like Nature, which is what we're hoping for, it's going to be on the radar of people who were never seeing hypermobile EDS. It's going to really put it out there even more than it has been in the past. But then our future plans of developing diagnostics and therapeutics have a huge potential to make a difference. Diagnosing children at a younger age and maybe getting them into physical therapy sooner, finding the right physicians early on, having those kids make lifestyle choices like, maybe not running marathons or things that could cause a lot more problems down the road. And then the treatment option is obviously the biggest goal here, is having something to go back to patients that can improve quality of life, other than, pain management and surgeries and frequent medical procedures, really.
Yeah. That was good.
I just all the questions. I wish I could. So I do want to, before we end, for those who had questions on the side that we didn't get a chance to answer, there are some really good questions I'd love to answer. Please drop me an email and I will answer them. My email is EDS registry.
I'm typing it.
Oh, you're typing it?
Yeah.
Oh, there you go. Yeah, so just drop me a line. And especially the question about the loss of function would be a autosomal recessive. I really enjoy talking about that.
Do you want to talk about it a little bit now before I ask the last question? You can.
Sure.
You might bore everyone. But...
It's all good.
Yeah, so in the genetic studies I've been involved with, the autosomal dominant, where we've identified genes that have just been monoallelic. So one allele of the gene was found to be mutated. So one copy. And so this essentially makes the person or the animal a heterozygote. Recessive would be a homozygote. There are certainly a lot of recessive diseases. The inheritance pattern for hEDS in our nearly, 500 families or whatever it is all autosomal dominant. So we know that the tracking of the disease does follow an autosomal dominant pattern. How does that work biologically? Autosomal dominant doesn't mean that the mutation is necessarily dominant. It can be a dominant mutation where it's achieved a new function to suppress something else. But in our case, in the case of the gene that we identified, okay, we identified an enzyme. The gene in the large family was an enzyme, and enzyme levels are extremely tightly controlled in the body. That 50% less of an enzyme, which is what we see in the patients, is a sledgehammer. And so simply losing one allele of that enzyme has really rendered the patient with some big problems. So it doesn't necessarily mean that a loss of function would be autosomal recessive. It can happen that way, but in this case, it doesn't. So,
That's interesting.
is it, Terry, did that answer the question? If not, we can continue the discussion offline. I would love to.
There's an email in the chat. And again, it's EDS registry at musc.edu.
Yep.
And now I'm going to end on looking forward. So there was even a question, would patients that maybe see Dr. Patel, they might be interested in like giving their sample to join this registry for patients who might be undergoing surgery even. So how can people get involved in the study? I left it up on purpose so that you can gesture to it. And then if there's any way that people can get in touch with you, I guess, what are we missing?
Right. So if you go onto our website, shown right here, or scan the QR code, or if you just Google like Norris Lab at USC, you can find us. There is on our website, you'll go to the top, it says research. You click on Ehlers-Danlos and it'll bring you to a page. At the bottom, it says if you're interested in enrolling in our research, you can fill out this survey. The survey is just a really short survey to make sure that you're eligible. So I don't know if everyone that's watching is in the US. At this time, we can only take people who are in the United States and over the age of 12 with a clinical diagnosis of EDS or hypermobile EDS or a family history of hypermobile EDS. If you've already filled this out and you have not been contacted yet, you will be contacted by a clinical coordinator soon. If you fill it out again, it's not going to make it happen faster. I know some people are doing that. They're going in the order that they get them. So they go through and call people or email you first, actually, to set up a time to call you. And then you'll get a phone call where you'll go over the consent and what's involved in the study. If you're trying to get in contact with us on the email I just shared, so the EDS registry at musc.edu, the email is also on our website. I also am people send me Twitter messages. That's fine. I'm on Twitter. I do my best to get back to people. I'm also on Instagram and I post about our research a little bit. Court does science on both of those. Chip doesn't really know how to do the social media thing. But I also do my best to get back to people as quickly as I can on there. And I do read, I am like Chip, I do read everything. Sometimes it just takes time. But even just like the words of encouragement are awesome and we really appreciate it. I think that answered all of it. Yeah.
Yeah, words of encouragement are wonderful. We do get some people who are, unhappy with us for not revealing what the gene is, and but it really needs to go through publication and needs to be peer reviewed, and that is the ultimate test, right? But I mean, that's so we can't reveal anything before that. And in fact, if we were to do that, then a journal could easily say, you've already disclosed this information. We're not going to publish your paper. So, I know people want to know, and I know it's important, and know that we're, working as hard as possible to do it. And
I totally understand that. It's so important that it goes through the proper channels, because if it doesn't, then what's tragic is that this breakthrough gets lost, and I would be so upset if that were to happen. So I'm really, as much as you want to keep hush hush about it, please do. And I am sitting here, I told you I was fangirling. If you got published in Nature, I would just like probably die for you. That would be great.
Yeah, that's like also every PhD student's dream. So it's double with the publication goals and working on something that's so important to me.
yeah,
It's no big deal to Chip to get published in Nature, but
I've had a few in Nature papers, so yeah, it deserves to be in Nature or Science.
Yeah, it does.
It does, and I think it will. I think it will go in Nature. So it just might take. It just might.
I
take
wish
a little.
you a very kind reviewer number 2, and we'll see how.
Yes, awesome. However, I don't think the reviewers I've selected have ever been chosen as reviewers. So, but sure, I'll put you down.
No, not well. If you want me to be reviewed too, well, it would be a very. But thank you both so much for this tonight. It was amazing. I'm very excited. I will be signing up to any RSS feeds to see when you get published, because I know you will. And everyone's thanking you. Thanks for your amazing work and commitment to EDS. Yeah. Thanks, Court and Chip. But thank you, and thanks everyone for joining us. Dr. Norris and Cortney, I'm actually going to send you this video when we get it all set, just to make sure everything's kosher, because I do want this published. So I want to make sure that nothing was accidentally put in here, so we can see it.
Yeah, I think we'll be okay. And Kathy and Kevin, if you're still on the line, drop me a line and I'll answer your question about the homozygosity question.
Yes, absolutely. Thanks, everyone. Have a great night.